At any point in our lifetime, we would have definitely visited the physician for medical treatment. Going back to our childhood, we can recollect a number of instances where we used to run around the house to find a hideout so that we are not forced to take the syrup or tablets during sick times. Definitely, the medicines did work and we were back to routine. But have you ever wondered what the medicine did to make us better again? We know that medicines are antibodies which fight against the foreign bodies such as virus, bacteria, etc., that enter into our bodies to make us fall sick. But how does it fight? Is it with the swords and warriors? No, but metaphorically, yes. Antibodies, along with its warrior and sword like antigens, binds with the molecules of antigen to win the fight. Now the question is, what are antigens? In this session, we shall discuss about antigens, types of antigens and structure of antigens. We shall also discuss about the importance and the significance of antigens. A substance that binds particularly to the respective antibody is called an antigen. The strategy of an antigen and an antibody is the lock and key model. During 19th century, in his side chain theory, Paul Ehrlich gave the term antibody. The term antigen was derived from the antibody generator. Our immune system produces antibodies once the antigens enter the system. Antigens may be foreign substances. Examples of antigens are chemicals, viruses, pollens or bacteria. Within the body, antigens may also be formed with tissue cells or bacterial toxins. The antigen may develop from the external environment or from within the body. Immune system usually tolerates self antigens. The self antigens do not react against the structures present in the body under normal conditions. Non-self antigens can be referred to as invaders from the external world. They may also be described as harmful or modified substances present below the disturbed conditions within the body, and thus these are imagined or guessed to be attacked by our immune system. In other words, our immune system will try to kill or neutralize any antigen that has been identified as a foreign substance and signal of harmed tissues. Through a histocompatibility molecule, cells present their antigenic structures to the human immune system. Several kinds of immune cells will be activated depending on the antigen introduced and the character of the histocompatibility molecule. Antigen is primitively a structural molecule that particularly binds to the antibody, but the term now refers to any molecular fragment or molecule that can be identified by extremely variable antigen receptors of the immune system. By major histocompatibility complex, say MHC, it should be refined into smaller fragments inside the cell and delivered to a T cell receptor for T cell receptor recognition. To elicit the immune response, antigen must need the help of an immunologic adjuvant. The adjuvant component's essential role is in the activation of natural immune system. When an immunogen, which is an analog to the antigen, is injected in the body, it will provoke the immune system. The immunogen is a mixture of substances which has the ability to initiate the first immune response. Later, the adaptive immune response is activated. Once a highly variable immune receptor products are produced, the antigens bind to them. The products are B cell receptors and T cell receptors. The ability to cause a cell mediated immune response or humoral immune response is known as immunogenicity. The ability to combine, particularly with the last products of the immune response, is known as antigenicity. Though all the molecules that has the property of antigenicity also has the property of immunogenicity, but the reverse is not true at all. An antigen at the molecular level can be qualified by its ability to be adhered by the variable region of an antibody. Different antibodies have the ability to separate between particular epitopes that are present on the antigen's surface. The structure of an antigenic epitope is changed by a small molecule known as hapton. The hapton molecule must be attached to a large molecule carrier, such a protein, in order to stimulate an immune response. Antigens are normally polysaccharides and proteins, less often also lipids. This also includes viruses, bacteria, and other microorganisms. Only when attached with proteins and polysaccharides, nucleic acids and lipids will be antigenic. Non-microbial exogenous antigens include egg white, pollen, and proteins from transplanted organs and tissues or on the transfused blood cells surface. In an immunogenic form, vaccines are examples of antigens, which are deliberately administered to stimulate the memory function of the adaptive immune system for the antigens of the pathogen capturing the recipient. In order of their class, antigens can be classified. Let us discuss about each one of them in detail. The antigens that enter the body from the outside world are called as exogenous antigens. Some examples are by injection, inhalation, and ingestion. The immune system's response to exogenous antigens is often subclinical. The fragments are processed by taking the exogenous antigens into the antigen presenting cells. They are APC. This process is done by phagocytosis or endocytosis. By the use of class II histocompatibility molecules, APCs deliver the fragments to T helper cells, that is CD4 plus cells. They get activated and secretion of the cytokines is started. Cytokines substances have the ability to activate antibody secreting B cells, cytotoxic T lymphocytes or CTLs, macrophages, etc. Some antigens are exogenous at the beginning and later they become endogenous. By distracting the infected cell, the intracellular antigens can be released into the circulation. Antigens that are generated within normal cells are known as endogenous antigens. The generation is the result of any intracellular bacterial or viral infection or of normal cell metabolism. If any triggered cytotoxic CD8 plus T cells identifies them, the T cells starts to secrete many toxins, induce the apoptosis or lysis of the infected cell. To prevent the cytotoxic cells from killing cells, self-reactive T cells are immediately deleted from the collection. This is the result of tolerance, which is also known as the negative selection. The endogenous antigens include autologous and idiotypic, genogenic or heterologous, and allogenic or homologous antigens. An autoantigen is generally a normal protein or a complex of proteins that is identified by the human immune system of the patients suffering from a particular autoimmune disease. Under normal conditions, these antigens should not be the target of the immune system, but because of various environmental and genetic factors, for such an antigen, the normal immunological tolerance has been lost. Tumor antigens are antigens that are present on the surface of the tumor cells. At times, these antigens can be produced by tumor cells and not by the normal ones. They are called tumor-specific antigens or TSAs in this case. Normally, they result from a mutation called tumor-specific mutation. Normal cells and tumor cells present more common antigens, and they commonly are called as tumor associated antigens or TAAs. Before the cytotoxic T lymphocytes proliferate or metastasize, they identify these antigens and may destroy the tumor cells. In the form of mutated receptor, tumor antigens can also be present on the tumor's surface. In this case, they will be identified by B cells. Antigens may be either polysaccharides or proteins. Typically, an antigen is specified as a substance that holds to the specific antibodies. The antibodies in the human body identify the potentially harmful foreign substances and neutralize them in the bloodstream. The particular binding of antigen and antibody is similar to the lock and key binding model. In human blood, particular antigen present in an individual's blood assigns the different blood types using different. The A and B blood types are determined by N-acetylgalactose or galactose monosaccharide, while all the blood types comprise the oligosaccharide or O antigen. Similarly, both A and B antigens are present in the AB blood group. To define the positive or the negative state of A, B, O blood groups, additional antigens are bound to them. The enzymes that bind to the antigen have similar structures and slightly vary by demonstrating the antigen specificity. Antigens are not only used in the ELISA technique to identify the presence of an antibody, they are also used for preparing vaccines to demonstrate or to improve the immunity to a specific disease. When the pathogen gets into the lymph and blood, antigens present on the surfaces of the pathogens induces the lymphocytes to develop or produce particular antibodies which destroys the pathogens by enhanced phagocytosis, lysis, holding the pathogen together, or by neutralizing the toxins from the pathogens. The primary infection involves a particular antigen. The responding naive cells multiply to develop a colony of cells. Most of the multiplied cells transform into effector B cells or plasma cells to overcome the infection, and the rest of the cells remain as the memory cells that can live and survive for years or even survive for a lifetime. A potential period for the production of antibodies is needed, for this is a complicated set of reactions. When the immune system tries to invade the pathogens, the already produced memory cells trigger the lymphocytes to develop a large amount of antibodies immediately. The antibodies developed in the primary response can be maintained for a very short period of time, and the antibodies developed in the secondary response can be sustained for a longer period. Vaccines are made to enhance the production of the memory cells and to get them ready to fight against the pathogens in future. Three types of antigen-based vaccines are produced. They are the purified vaccines, recombinant vaccines, and synthetic vaccines. Let us get to know about each of them in detail. The purified vaccines are also called as subunit vaccines. They are composed of small fragments of molecules that are purified right away from the pathogen that yields a protective immune response. The purified vaccines can be polysaccharides, proteins, or exotoxins. The examples are, in children, vaccines used against causative agents of meningitis. The recombinant vaccines are immunogenic proteins that are being produced by the genetic engineering method. DNA that encodes for an immunogenic protein of a pathogen molecule can be inserted into yeast, bacteria, and viruses that infect mammalian cells. The proteins produced endogenously by the cells and can be harvested. Example, hepatitis B virus surface protein, simply called as HBsAg. synthetic vaccines are peptide antigens produced by automated machines. While the synthetic polysaccharide technology is under development, the synthetic polynucleotide technology already exists. Thus, some knowledge is required for the conformational structures for B cell epitopes and for anchor residues for the T cell epitopes. The B cell epitopes may be sequentially assembled. Though computer algorithms are available to help in selection, trial and error approach is still being needed. The characteristics of a good antigens are the structural stabilities area and the chemical complexities area within the molecule are required. The lacking of extensive repeating unity in significant stretches. An antigen should have maximum molecular weight of 8000 to 10,000 Dalton. An antigen should have minimum molecular weight of 8000 to 10,000 Daltons. Although it happens in the presence of a carrier protein with molecular weights of 200 Dalton have also been used. An antigen must have the ability to be processed instantly by the immune system. Antigens must have immunogenic regions that can be accessible to the antibody forming mechanism. Structural elements of the antigen must be different from the host. In peptide antigens, at least 30% of the regions must contain immunogenic amino acids such as lysine, glutamine, arginine, glutamic acid, asparagine and aspartic acid. Significant hydrophilic or charged residues must be present in peptide antigens. To summarize, an antigen is a substance that stimulates the yielding of specific antibodies that reacts with the antigen. Various antigenic substances are present in viruses and microbial cells. Specificity is the most significant feature of antigen-antibody reactions. The antibodies formed as a result of immunizing an animal with one microbe will never react with the antibodies that are formed by immunizing with the different microbe. In the blood serum of animals, antibodies can be found, and using sera, the laboratory test of antigen-antibody reactions are carried out. A microorganism can be characterized by its antigenic makeup, and also microorganisms can be identified by using different serological tests. Antigens and antibodies are important features of immunity, and the immunology is added in the science of microbiology. An antigen is molecule when introduced into the human body triggers the development of an antibody by the human immune system, which will then kill or nullify the antigen that is identified as a foreign substance and a potentially harmful invader. These invaders can be pollen or cells such as bacteria. The term was originally derived from antibody generator and a molecule that binds particularly to an antibody, but the term now also refers to any molecule or molecular fragment that can be bound by a major histocompatibility complex or MHC complex and delivered to a T cell receptor. Self-antigens are normally tolerated by the immune system, where non-self antigens are noticed as intruders by the immune system and attacked. From the immune system, autoimmune disorders arise reacting to its antigens. A specific type of antigen known as immunogen is a substance that is able to evoke an adaptive immune response if injected by its own. In another way, immunogen has the ability to induce an immune response once they are made. The antigen molecule is able to attach with the products of an immune response. The concepts of immunogenicity and antigenicity are different. Through histocompatibility molecule, all the cells show their immunogenic antigens to the immune system.